latest SFBigAnalytics event. Welcome. We are just over 9,000 members, and I'm super excited about that. So this is awesome. It's because we keep bringing great people to talk, and tonight's no exception. Before I introduce tonight's speaker, I just want to mention our next meetup coming up on April 18th at Yelp. For those of you who are into like deep learning and biology, there's a cancer detection talk using deep learning, as well as we're going to get some inside scoop on GoPro and how they build their analytics in the cloud. So that's next time. All right, on to tonight. the Wi-Fi password.
123 GP GP GP.
It's the guest password is 123 GP GP GP.
Lowercase.
Lowercase.
Thanks to GoPro for being awesome and hosting us tonight. So, our speaker tonight, I was just floored when I saw how many citations his papers have. I had to update our meetup. He's got over 40,000 citations for his papers. And our speaker tonight hails from lots of different fields. He hasn't just gotten education in different fields, he's mastered so many different fields: computer science, mathematics, evolutionary biology, biochemistry. And you might think, how could someone master so many different fields, and how could they combine all of that expertise into one talk? What? So you're going to see tonight, Dr. Mark DePristo hails from, his pedigree includes Northwestern University, Cambridge, Harvard, and for those of you who are geneticists, the Broad Institute, you'll be very happy to know that he was running their medical and population genetics team there. Right now, he's over at Google Brain, heading up their genomics research over there. And tonight, we're going to hear, you can read the title for yourselves. Please join me in welcoming Dr. DePristo.
Thank you for the invitation. It's a real pleasure to come and talk to people, especially a diverse audience with a lot of different backgrounds. So, my hope with this talk is really give people a chance to sort of hear about our work. We can chat a little bit. I'm happy to take questions and just talk through the material. There's a lot of material in a whole bunch of different flavors. So there's stuff on deep learning, there's stuff on technology, there's stuff on mathematics. We can jump around as people see fit. If we go through really quickly, maybe we'll get through all the material. So, but that's not a goal. There are thank you slides in multiple places in case we have to stop early. So, what I'm going to talk a little bit about is some of the things that I've been doing for the last 3 years while I've been at Google, trying to understand how to use deep learning technology in biology. And so I run a team now in Google Brain whose mission is largely to find problems that look like we could do deep learning and actually figure out how to make them work. And so this is one story about that, and then the slides sort of end with another story about how other people might be able to do that. So I thought I would just introduce, the taxonomy of deep learning and machine learning. And these are interesting fields. There's a lot of buzz on deep learning, but it's in a long trajectory for a very long time, and it's much less mysterious than it seems to be. So roughly, the taxonomy of machine learning kind of boils down into two fields. There's like AI and machine learning. And they're roughly defined in terms of making intelligent machines versus making machines that learn from data. So those are like the fundamental difference. So we had this vision that we would someday sit down and program a robot that could do everything we do, that it didn't work out so well. That turned out to be a very hard problem. But we made a lot of progress in another field of teaching machines how to learn from data. And it turns out that with enough data and enough sophistication, you can kind of get into things that start to feel like an AI, even though it's not really doing anything but learning from data. So examples here is like IBM Deep Blue. This is the machine that plays chess. This is really classical AI approach. You have a giant rule system, you have a really powerful computer, and it churns through as much data as possible to beat you in chess. On the other, couldn't be further away, is like ATM check readers. So this is a machine learning problem. You take photos, and then you've got to read out the little digits with those little barcodes. Nobody would claim that's an AI. Nobody's afraid that that's going to take over the world. But there is sort of an intersection that goes from, it starts in ATM check readers and kind of goes into AlphaGo. Now AlphaGo is a program that's super sophisticated. It learns from data alone how to play the game of Go, and Go is a very challenging game. And so if you have enough data and you can learn very efficiently off that data and very effectively, you can teach systems to become kind of artificially intelligent, to the point that they can do things that are actually kind of outstanding and amazing even as a human being. So, another way of thinking about that is, how do you solve problems? This is sort of a nice introduction. Again, I have lots of beautiful slides that I've stolen liberally from other people at Google. So these are Google caliber introductory slides. So one way of thinking about these challenges, like how do you write a spam detector? Very standard, very general problem. Everyone's familiar with it, although less and less now, because spam is pretty well solved by these systems. There was a day when spam was a huge nightmare. And what ended up happening, as soon as it started showing up, because nobody anticipated spam, you start doing what you might realistically do in that scenario. You sort of say, okay, well, I'm gonna make some rules that like filter spam and non-spam. And you write a whole bunch of kind of rules that look something like, does the email contain Viagra? And one of the challenges with this approach is sort of exemplified in the spelling of Viagra here. Like I'm not even sure how many people saw that it was spelled like that. Because you can just read right through that kind of characterization of the word. But a program, generally, if that's a string in a programming language, that's not going to show up as the Viagra string, right? It's got some explanation point, it's got a non-standard A. So you start to get into this sort of arms race between the spammers and these rule systems, right? The spammers say, oh, I can fool the system by replacing I's with exclamation points, or I can add a space, or now it's like Unicode characters that do all sorts of crazy things and spell out words. How do you fight that? So what you end up having is, in a rule-based system, like an endless series of these sort of tests. You get a giant database of rules, and to the point that you have like public rule sets for spam, and everyone uses that. And it turns out that doesn't work so well, because you're much better off learning from the data. So modern spam systems are built like this. You have some system that can classify emails that sort of walks through whatever's there, text, images, the headers, everything, sees all the data, and you're going through manually and saying, this thing's spam, that thing's not spam. And you're demanding that the computer learn how to classify the two. It learns how to differentiate between spam and non-spam. And the beautiful thing about this, and the really interesting algorithmic problem, is how do you change the program so that it gets better? Like that's really what machine learning is all about, improving the program itself so it does better at its classification task. And the cool thing about this, ultimately, is it changes the problem from trying to write a whole bunch of clever rules, which become very, very challenging to do, to very general problems, like how do you build a classifier that can learn from data, modify the classifier based on feedback, and collect data and label it. Right? So that you enter into sort of a virtuous cycle. The more spam that arrives, the more spam you can label, the more effective your systems get at identifying and separating the two. And at some point, it becomes so good that it's almost impossible to send spam through, which is kind of the state we're in today. So I think it's pretty rare to get spam right now, but 10 years ago it was not. So what is deep learning, right? So deep learning is sort of the modern incarnation of that change the program and improve it. And it turns out to be very, very simple. At the end of the day, there are these simple neural networks, which are programs that really can do this kind of problem, classification, and look at data. And what we learned to do in the sort of 80s and 90s is how to do shallow neural networks. So what this means is that there are not a lot of layers. So you can think about it as a shallow neural network is some kind of simple thing that takes an image and it does, one or two simple mathematical transformations of it. And then from those mathematical transformations, you're forced to make a statement, like, is this a cat or a dog image, or is this spam or not spam? What deep networks are obviously what the name implies. They're just very deep versions of these networks. There's really nothing fundamentally new about them, besides that they're very, very deep, and we have effective engineering tools that let us train them, which is really the triumph. So, in many ways, deep learning is like the triumph of engineering over science, because nobody really understands why it works, but it really works really well, and it really scales out super well for big amounts of data. So it's sort of pragmatically useful, but we're still trying to figure out why it works at all. So what we're doing in a deep neural network is straightforward. We take this picture of a cat. It's a bunch of numbers. This thing is a width by, usually it's a width by height by 3-dimensional tensor. Now it's this abstract representation. And what you do is you have some transformation that goes from like some shape of input that looks like width by height by rank tensor, and turns it into another one. I might change the shape. That's sort of illustrated here. It's changing the dimensionality. Sometimes they're deeper, sometimes they're wider. And all these transformations can be different. in many cases, the really important thing is that transformations have non-linearities in it. But suffice it to say, like, you just basically string up a whole bunch of these things together, and you demand that it learns how to call cat when its input is this. And the machinery that's sort of hidden away here in these gray boxes is how, when it says dog, how do you make it better at recognizing cat? Like, how do you update the transformations such that when it makes a mistake on dog, and it should be a cat, how do you update these transformations such that it gets slightly better at recognizing cats? And that's really the fundamental kind of optimization problem of deep learning. But this is kind of deep learning in a nutshell. If you have these really deep networks, they're composed of like relatively simple transformations that are sequentially stuck together. And because they're constrained to have particular properties, we know how to train the networks that are very, very deep effectively. So what you do when you have such a really deep network is you collect lots of data, train, and it gets very, very good at differentiating cats from dogs. So that's sort of fundamentally what's happening. This is another way of thinking about it. This is actually a pretty challenging picture of a dog. It's not obviously a dog. I can tell you it's a dog, and we'll agree, but it could pop up and be something else, right? It could be a capybara, for all we know. So what's really going on at the core of this, after you've trained this thing, there are these so-called activations. So what's going on, and I mentioned in passing, there's sort of a non-linearity in most deep learning networks. What that really means is that at this point, this pixel is activated because its value is like above 0. And if it's below 0, you get nothing. So you can think about it as like, once you pass some threshold value, it starts to emit a value out of it. Otherwise, it's just emitting 0. And that's why you think about it as an activation. It's like looking at its inputs and deciding whether it should be kind of on or off, and typically with some kind of a scale associated with how much on it is. And so that what ends up happening when you train these networks is like the input comes in, there's a dog, like the activations start to capture the properties of being a dog, and then it kind of goes up and up and up as it refines its understanding of like this thing is a dog. And basically at the end, these last activations are essentially statements about being a dog or a cat, to the point that the last value is literally translated into cat or dog. So it's like a series of things. And at lower levels, it's doing things that like your brain does in visual processing. So it's recognizing like rough shapes, like it's semicircular, like dogs have hair. Like these sort of properties are learned at lower levels and composited up until it's like, okay, look, it's hairy and it's semicircular and it's not, there's lots of properties that it learns. Like it has 2 ears, it has eyes, and it slowly goes up and until the point it's like, okay, this thing is a cat or this thing is a dog. So that's really it. Like that's the magic of deep neural networks. You can certainly go get Ian Goodfellow's book and read it and learn lots more about the technical details. But fundamentally, as a engineering methodology, it's just a series of simple transformations. There are systems that can train these systems of transformers to basically recognize interesting things. And you can expand it out and do all sorts of cool things. And the reason people love these is that they're extremely general. Like you can download TensorFlow from Google and solve like cat versus dog. You can also tell it to translate, English to Chinese. It's the same kind of fundamental structure. And the whole, and because it's all engineered really well, like you can kind of treat problems in the same way, which is fascinating. So an example of this is things that we know work. And this is why everyone's excited by deep learning, because it works in so many domains, and it really does very, very well. So you can do the cat versus dog, if you have an image. But you can do spam. This is how spam works in Google. You can translate audio signal into text. So you can do translation, and you can do crazy stuff like this. You can demand that it emit a caption on the image. So these are actually automated captioning systems. So you take a picture and you say, give me some kind of description of what's in that image, and it says a close-up of a small child holding a stuffed animal. And that's like shockingly amazing. So, everyone says, oh, I'm pretty good at cat versus dog, but this is actually genuinely in the realm of AI now, I would say. And the thing that's actually what really blew my mind in the way you build this system is this is actually composed of two deep learning models stuck together. There's a model that recognizes what's in the image, and there's a model that does language translation. And if you stick those two together, you can get it to emit things like sentences with captions. But it's fundamentally composed of like a cat-dog recognizer and something that looks like a translation system. And you jam them together to co-train them, and then they can emit captions, which is kind of mind-blowing. So that's sort of my introduction to deep learning. Obviously, it's not super technical. It doesn't explain how to get it to work or anything, but it's sort of enough, I think, to set the stage of why we think this is interesting. So this is a good time to talk about deep learning before we go too far into genomics. If anyone has questions, or clarify. Okay. Good. So I run the genomics team in Google Brain. We do kind of 3 or 2 things, depending on how you count them. So the 2 big items are, we're trying to make TensorFlow work really well for genomics data. So it turns out that in TensorFlow, if you're an image or a text sentence or you're a spam email, it's very, very easy to work with that. The deep learning systems were developed in many ways on data types that look like that. So they're natively very, very good at handling that type of data. It doesn't really translate super well to genomics data. So we spend a lot of our time trying to shoehorn genomics data into TensorFlow. And deep variant, which I'll talk about a little bit, is the most egregious example of shoehorning things in. So one of our missions is to stop having to shoehorn everything in. How do we build models? How do we read data that's native to biology and have TensorFlow work well on it? And then we do the other side of it. It's not always obvious how to build such libraries. It's not really clear what's important and valuable. So we do actually a lot of work on trying to apply deep learning tech to kind of bioinformatics problems. So we look for things that are, we try to say, has anyone done this? Is it interesting? How important or impactful would the results be? Just to kind of show people what's possible in the same way that AlphaGo is super exciting and motivating for people who have planning problems or learning from data. It's very accessible. So we try to do the same thing, but in the kind of life sciences space. And we like things that have broad adoption. So we tend to go after kind of infrastructure level stuff. So just getting the nice scientific paper out isn't really our goal. Our goal is to have broad use. And then we do a lot of stuff like open sourcing and other things. We come and give talks like this. So we want people to sort of get excited about genomics as an interesting area for deep learning. So transitioning right into our program, Deep Variant, I sort of tell you a vignette about an actual problem we solved with deep learning, about what it does, how we did it, why you'll see the shoehorning in a little bit, and I'll explain why it's not so weird. So I'll just start with now we have some biology stuff. So I'll just, who here has actually heard of genome sequencing? Okay, good. Oh, okay, that's good. We'll go fast then. So genome sequencing is exciting, mostly because everything has genomes. So it's like universal technology. You can go measure the genome of a bee, you can measure the genome of a virus. This is a picture of my long-term collaborator, Ryan Potlin. We can go measure his genome with this instrument. Since everything basically has genomes, we have a universal tool to study the genomes of all organisms. And because of this universal tool, and because it's pretty inexpensive, there's a lot of work that gets done with sequencers that are super interesting. Everything from finding the genes associated with schizophrenia, which is a super important medical problem, to studying this genome sequence of Neanderthals by digging up bones and sequencing them, and tracking things like the outbreak of E. coli at Chipotle, which is all done now with next-gen sequencers. So this is a very general technology. It has lots and lots of applications, so it sort of fits into our broad use goals. The thing about genome sequencing is not so old. I think it's, sequencing's won at least 2 Nobel Prizes so far. It turns out actually the sequencing of proteins started before DNA. It was slightly easier. The DNA sequencing started and it became this runaway success. The Sanger sequencing sort of was out here. We solved the structure. We solved the first sequence of a human genome out here. It was very expensive. The first one was $1 billion. A lot of work. This is a major, major effort. Of course, that's sort of disappointing when you want to sequence billions of people at billions of dollars a genome. This is super intractable. So the problem is, although we were following Moore's law on the cost of a genome sequencing run, we're still, if we followed that original curve, it would be 2015, and we'd be talking about how excited we are to have a sub-$1 million genome, which is not so exciting. But there was this inflection point where we really went on a different curve. And that curve was the introduction, that inflection point was the introduction of so-called massively parallel sequencing technology, which, to a first approximation, actually just trades quality for quantity. So you figure out a mechanism for measuring sequences at very high scale for very cheaply, and that's super exciting. We're willing to give up a huge amount of quality in exchange for how cheap the data is. So at this moment, the data got cheap, and it got super bad. And it was really abysmally bad. So what actually happened is, just to be clear on these instruments, our genomes are like 3 billion DNA bases long. They're in 23 contiguous chromosomes. We have 2 copies of each. Your fantasy instrument would like read off each one of those in perfect fidelity, and you say this chromosome 1, which is 250 million bases long, looks exactly like this. This is not at all what you get. What you actually get is a billion 100 base pair long DNA sequences, which are tiny, and they're from random points in the genome, you don't know where they are. And you have huge base error rates. So the things you're measuring have error rates that charitably would be 1 in 1,000 bases are wrong, and frequently are up at like 10% of your data is wrong. So it's super errorful. And to make it worse, it's highly correlated, the errors. So it's not just random errors. It's like, the errors are related to the DNA sequence itself. So the thing you're trying to measure introduces specific errors based on the thing you're measuring. So it's super difficult. So the way to think about this is a nice image from Pavlov-Pesner, talking about, imagine trying to do genome sequencing, but now you're doing it on books. So what here, this is New York Times article, but you take a stack of a bunch of New York Times, you blow them up, now you need to reassemble them, and then you can want to ask questions, like at some point, on some day, on some page, like exactly what was said. So this is a pretty challenging kind of algorithmic problem, because the data is big. I mean, there's 3 billion bases total in size, but you get to measure 1 billion 100th base pair fragments with tons of error. And you got to reassemble this thing and answer questions like, given what I just measured about your genome, what's your chance of having schizophrenia? So this is not these are not just fun questions that people want, are happy to have a little bit of error. Like, that's a serious question to ask. You need to do like a high quality answer. And it can't be like, well, I'm pretty certain that you have this genetic variant. It might not be there. Like, it's unacceptable from due to the medical implications of what we're doing. So the standard way we do this is, we bought this very expensive reference genome from humans. It's like $1 billion. We better use it. So we take these billion reads and we align them like strings to the genome. Because I have a pretty good picture of like what the 3 billion bases look like exactly. And what you do now is you're kind of left with a problem that doesn't look so daunting. Sort of, at least it's a little simpler. You don't have a billion things of unknown location. You have like rough string match-based locations. And you get piles of data. Like here's the reference genome. It's giant and contiguous. And then you get these little reads that are sort of matching everywhere except for a couple of spots. And the spots that are different places where you differ from the reference genome, because everyone differs at roughly 1 in 1,000 bases from the standard human reference genome. So what now you go through is you say, oh, I know how to handle problems like this. I have like multiple points of evidence. I can use like normal statistical techniques to decide like how reliable is that G. So if I only have one observation, maybe it's one of those random errors. I have 3 here. Is that real? Is it real enough to like tell you that you have elevated risk for schizophrenia or Huntington's disease or cystic fibrosis? Like these are major concerns. So how much data do you need? How much evidence? Like how certain are you? There are big ticket issues here. And then what you do is once you figure out what the real differences are, it's pretty straightforward to sort of think about it as a diff from the genome, and then you go and diff the genome and have a full genome sequence for an individual. So it kind of makes sense. I kind of led the story of like, oh, hey, we need to think about evidence. We need to think about how to do statistical incorporation of information. And that's really what a lot of things look like. So if you go out and you buy like your whole genome sequence from Illumina, and they give you a big pile of reads, and you align it to a genome, like you can go run a whole bunch of different types of tools of varying degrees of statistical sophistication. There's a section of the talk where we'll go into like painful detail on what those details look like. But just at a high level, you could say like, what do you have to deal with if you're going to try to solve the sequence of the human genome from a big pile of those reads? Worst case scenario is the DNA itself could be bad. So that would be like, maybe we're studying Neanderthals, and they didn't preserve their bones in the most hermetic environment. They died in a cave 50,000 years ago, and we're digging it up, and it's been in there forever. Like, things are eating it, like it's a nightmare. The DNA is very degraded, so like the DNA itself could have tons of errors. How you put it on the sequencer can matter, like what happens to the sequencer. Like at the Broad, there was a story, actually, at one point we could tell where the sequencers were in the room, because I could tell how far it was from the air conditioning vents. because you could see that the reactions were happening at slightly different temperatures. So introduced all sorts of air circulation and everything, but the actual physical properties of the instrument, exactly where it is, in a room, it's actually kind of a problem here in California because of the frequency of low-grade earthquakes. So it's like actually harder to sequence in California than it is in Massachusetts. And you have all these data processing artifacts. People have bugs in their programs that you can't overcome, etc. So how do people do this? And I was involved in the creation of the tool called the GHK by the road, and we kind of threw the kitchen sink of statistical tools trying to deal with every source of error. Right? We have a lot of data. I mean, the nice thing is every sequencing run produces 1 billion observations. So I have, for $1,000, I get an enormous amount of data. So I can build very sophisticated models very quickly, like trying to say, oh, if I have an A followed by a G followed by a T, the chance that the T is an error is 5%. If it's followed by a C, it's actually 10%. I can build models like that. So you build very complicated systems to try to kind of back out all the error properties, and you fit them all and then use that statistical approach to say this is good or this is not. The problem is when we did that, like many kind of big pipelines of statistical tools, everything kind of is handcrafted. There was a time when I was looking at so much of this data, I was like, oh, I should implement this type of transformation of the data, and I'll count in this way, and I'll have this threshold over here, and it's very hard. Like, it works really well for the data set that you have, but when I did that 8 years ago at the road, like, who's going to update those parameters? Like, how would they know? Like, now we have not 3 sequencers, but there's like 20 different companies making sequencers, each of which has their own trajectory of development. So you have to parameterize each one. You can have 100 people doing that. It's crazy. It doesn't scale. And the problem is it requires domain expertise, which is always rare. Like, there's not that many people who have like lovingly modeled sequencing data error properties. And I'm sure that's true in like every domain that everyone works in. Like, at some point, there's like one person who really knows what they're doing on your specific data type. And it's like, that's really hard. That's a difficult situation to be in. And it's really fundamentally difficult to generalize. So we were sort of many of the people on my team were involved in the creation of the earlier tools. We were very aware of like the challenges that we had, and we were interested. Honestly, when I joined Google, I was somewhat skeptical of this deep learning stuff. A lot of people walk around talking about like how machine learning is going to transform the world, and they do that every 5 years. So people, 10 years ago, they were like, oh my God, these SVMs, they're going to change my life. It's going to solve everything. And you're like, okay, like I'll try that out. It's okay. I mean, it's good, but it's not like transformative. And so, I was joined Google, and Google's telling me, deep learning is a big deal. Like, you should look at it. Okay. So I wanted to choose a problem that like I was very, very familiar with, that I knew was incredibly difficult. This is a really hard problem. It's lots of data. Big, you have to be able to do a lot of compute, and the error processes are super complicated. So you're like, okay, maybe this is a good fit, but also I have a very good sense of like what the baseline is. Like, how well can you do if you do everything you can to make it work well? Because I did that before I joined Google. So we said, okay, well, how would we recast this problem as a deep learning problem? And so what we did was pretty simple. That CG image where we've aligned things, this is sort of the generalization of it. What you do is you walk around through your experimental data, and you find all the sites that maybe have something interesting. So it's like, okay, at least 2 bases that don't look like the reference. And you tag it and say, this region is interesting. We're going to look at it more. You go through, you sweep through the genome, and you find like 5 to 10 million such sites out of 3 billion. So it sort of reduces the data down a lot. And then he said, okay, well, we don't want to build all these handcrafted statistical models. Like, what would you do? In the deep learning world, you just sort of jam all your data in some tensor and go. So that's what we did. We took literally all the data that the sequencer is emitting and just started jamming it into like parts. And originally it was an image, and now it's really a genuinely tensor. But we literally encoded this as an RGB image. We're like, okay, like just start stuffing it in. It doesn't matter. Like, let's let the thing sort it out. And then we just said, okay, we'll train a convolutional neural network and demand that it tell us how many copies of this C are really there. Is it 1 or 2, or is there none? And that's what we trained it on. It turns out this works super, super well. It's like shockingly, like amazingly well. Go ahead.
I have a question on the last slide.
Sure.
Or 2 slides ago.
This one.
So yeah, so for the center column, you're so aware of the drawbacks. Do you have a standard where you say, okay, this is the drawback that I know is going on with the existing tool set. Mm-hmm. Do I have a standard that I want the deep learning tool to meet? It's just that I know I'm happy with it.
Yes, I'll talk quite a bit about how we evaluate this. It turns out to be super interesting, and one of the reasons we chose this problem because there's really an unambiguous, third-party, vetted way of adjudicating which, how performant are the tools really. And I have to say, like, even despite that, people still say, I don't believe that it works. It's quite amazing, actually. It's quite an uphill battle. So I'm glad that there's such high-quality ways of evaluating in this area, because we're working in lots of areas and they don't always have such high quality standards. And that battle is super difficult. So what we do, and it's about to bleed into the answer of like, how do we know? So the goal here is these are what the images look like of the data when they were back in being RGB images. And you can see things that are happening even. Like this gap, because it's black, is because this person has a part of their genome deleted. So it's missing. And those are the gaps. Because you have a gap string alignment, you see that it's shifted over and there's 3 characters missing, so you get this consistent gap. And you're training the machine, you're saying, okay, given all this data, like predict the probability of these 3 states, homozygous reference, heterozygous, homozygous variant. And you demand that it do that, you send it into TensorFlow, and you say, okay, figure it out. It turns out to work really well. And I'll talk now about like how do you get training data, and the training data is sort of the inverse of the evaluation data. So it turns out there's this project in genome sequencing called Genome in a Bottle. This is administered by the National Institute of Standards and Technology, NIST. So this is such a big deal that like NIST has created a genome in a bottle for deciding how well, say, a sequencing lab is performing. So the way it works now is like you say, you have to go buy this sample from NIST, you run it, and you say, okay, how well did you do? And you have to compare kind of to their data for this person. And there was originally one person, NE12878, which I'm very happy to say I actually chose. I knew nothing really about this field, but I was like, this is the first sample that was really available to us at the Broad, so I chose this one and it became the standard. And there's 2 new ones. This is why the naming convention has changed, because this sample is so old that like we can't really use it anymore. It's an individual who was like semi-consented before like the ethical systems were put in place, so like it's quite a bad situation really. And then, so the newer samples are like beautifully well consented. But the view in the ethics community is that this person's already been identified. There's no additional harm and she's already dead. Which is sad, but she's been wildly influential in the area. So she's the best known genome in the world by far. And there are 3 of them now, and they've gone through these genomes and sequenced them kind of with every sequencing technology you can find. So you don't just do one Illumina sequencing run, you do Illumina, a PacBio, and an Oxford Nanocore, and a 10x thing. You do this Genomics. And you keep going and going and going, and sort of use this adjudication of multiple technologies to say this person, all these different sequencing technologies agree about what's in this region, therefore that's the answer. And they have quite a sophisticated now automated approach to doing that. And so it gives you a very high quality genome. And these numbers are continuously updated. I think now it's 95% of the genome is like considered like really known. But the thing about that is, what it really means is that we can take genome in the bottle samples and use them to train deep learning machine models. Because we can get data for this individual, make all those candidate calls, and then go label them because we know what the truth is here. And so this is how we feed and train the machine. And the inverse is that you can use it to benchmark. So we have quite a rigorous standard on how we do our training now. So we train only on 2 of the 3 samples. We leave out 1 completely, and we never train on the 20th chromosome of anyone. So like we can evaluate deep learning models on chromosome 20 for everyone that we train on, and we leave 1 kind of perfectly held out sample. So this is how we benchmark. And the reality is the numbers are, we're playing in kind of 5 to 6 nines. So we really care about the inner details of all of this stuff. So we're like, we want to make no errors in the genome, and we're like at 2,000 or something. So it's quite exciting, but it does mean that we care enormously about not overfitting. So how did we know this works? So what ended up happening is we were building this system at Google. We started building it in August of 2015, and they announced this Precision FDA challenge. So the FDA sponsored a competition on one of these samples and said everyone can submit calls for them, and the calls had to be submitted for NA12878, who's our training sample, and this HG002 sample, which they had yet to release. So nobody had ever seen the truth set for it. So it was almost a perfect evaluation. And it goes to the question that was asked of, like, people still complained that we were biased. And like, how is that possible? Like, the sample wasn't even publicly available. But so this is the first unveiling of the HG002 Ashkenazi male sample, whole genomes, lots of submission, because there's lots of interest in this area. And we won this, so very quickly we were the highest performing tool. So we worked for 5 years on the GATK, and in 6 months, 2 of us sort of easily beat its performance in the first go. And there was another one of these, the Hidden Treasures Challenge, which we again scored like the highest performance on the SNPs. And the second is on indels. This was to see, you can see the growing number of submissions, the growing number of groups who are sort of interested in playing in this space. This is sort of a real data slide in recent. These are old numbers, so we're up very close to the 100 and 100 on both sides now. But you can see there's interesting things that are happening. So like the GATK, these are 27 replicate sequencing runs of this sample. So verily, we sequenced this person 30x 27 times and just ran the tool in the standard way on it. And we wanted to see if there was more scatter, if there was more accuracy as well as less variability. And so you can see that these deep learning machines are sort of not only performing well in one case, but like you can just kind of do technical replicates, and it's more robust to variation in technology than a competitor tool. Go ahead.
So which was the first and which was the last run?
That's a good question. I don't know. Are you saying like is that a line? I don't know. It's a That's a great question. Should look that up. It'll be super interesting. So another thing that we wanted to ask was, it's one thing to be able to do very well for one sequencer. So we only got one sequencing data type for this thing. So we said, well, we don't really know anything specific about Illumina sequencers. So can we train it for other sequencing types? So instead, here you just go. We just went to the Genome in a Bottle data, website, download a whole bunch of different types of sequencing data off different types of instruments, train the variant to call variants on them, and in all cases, minus this one case, which is due to a technical issue, we vastly outperforming the other tools. For the sequencing, the new instruments, where typically our competitors are chosen by the maximal performance that they can achieve. Go ahead.
So what's the limit of detection for the SNP? Is there a requirement for the coverage, the minimum coverage?
It turns out, so yeah, I don't think I have this slide. It turns out that we actually are super good down to kind of 2x or 3x, where you have almost no data. As long as you show it, data of that form, it actually performs very well. So
There are 3 reads. There are 3 reads
covering a location, and 2 of them containing the reference and 1 contains the mutation. So 1 out of 3.
well, we can't do
And
any.
then no label.
Yes, I would say we can't do any better than our candidates. So our candidates kind of demand at least 2 alternate bases. But once you get to that, we actually have much higher F1s than anybody else, even down at that level of coverage. That's only true if you train it to see such low coverage data. But we were actually shocked. We were convinced that it would not work down at that. But when we run it, it actually does a pretty good job. So
It's really very challenging when the coverage is pretty low.
yeah,
So
so
wondering on the precision FDA, the challenge was the lowest.
the coverage is very high there. The
Yeah,
precision
yeah.
FDA is like
Yeah.
50x. It's very,
That's what
super high quality sequencing data. They took all the sequencing runs they could do and chose the best. Right, so it was like the maximally performant.
Just another question. So if there is a SNP combined with the indel on the same location, what's the performance?
Generally not. We were typically very, very worried about these cases. There are situations just to be, so there are, you can think about it like this. There's regions in the genome where you have simple changes. And this occurs like 1 every 1,000 bases. So roughly 1 every million bases, because it's the square of that rate, you have 2 changes that happen at the same site. So it gets pretty challenging, because now you have 2 independent events that kind of occurred at the same point. Like, can you see both changes? And it turns out that it does pretty well, even in that scenario. It turns out actually the scenario that it didn't do very well on was typically when you have lots of things in a row. So if you have a lot of differences close together, it found that more challenging to differentiate errors from non-errors. So it was pretty interesting in the details. So we started doing other things with this that were pretty interesting. So one thing that we did, and again, it goes back to kind of why do you do deep learning? Why, the challenges of handcrafted rules and things. So one of the challenges is, the official genome build of the human genome is going up and up and up. So we're at build 37. Well, we were at build 37. We're now at build 38. Every time they do this, they refine the genome, they fix some errors, etc. But it moves around the bases. So like a base that's 10 million bases in on chromosome 1 might be 9 million bases in on B38. Sorry, I should say that again. You can move around, so you could be at 10 million on B37, and the same base is actually at 9 million on B38. So it moves around the things. So what that means is that many of the other tools kind of need to be reparameterized every time the genome build changes. Because they have all this data they use, and that needs to be upgraded and mapped to different versions, all like these annoying technical details. However, it took years for any other program to sort of move from B37 to B38. Because nobody really wanted to do that work. It was really a thankless task. So for a long time, nobody could call anything on B38, even though it had been out for years. So we asked a very simple question, like, suppose I train something on B37, like how well do I do just calling things remapped to B38? And it turns out you do about as well as you do if you remapped everything to B38. So it's able to generalize across genome builds, which you would expect the machinery to do. It kind of doesn't know about the position in the genome, but as a matter of engineering value, it's actually super nice. Like it doesn't really need all this supplemental data. It sort of learned everything in this model, and the model is highly portable. And the most extreme example of that is you can train on human data and then call mouse data, which is a totally different organism. Because it doesn't need any resources related to the genome. It's learned everything about the human data, about how the sequencer behaves, and then you just call mouse. And this turns out to work very well on all sorts of different organisms. This is actually one of the most interesting things that deep learning can do. So this is an example of a work that we did recently on a whole bunch of different rice genomes. So here we're training, all this stuff is trained on human data. And we were asking, there's a big international effort to sequence all these different rice strains. These rice strains are inbred. So really the answer, although they have 2 copies of every chromosome, they really, all the differences should agree. So they should be always homozygous. There should never really be any differences where you have 1 copy of the difference and one chromosome doesn't, because they've been inbred. And so this ratio of how many hets to homozygous variants is a pretty good measure of accuracy, because every het is probably an error. So you really, really want a low rate. And this gives you a sense of the existing data set released. I mean, this is what people are working on, trying to understand the population genetics of rice, has an enormous number of errors, and if you rerun devariant on it, this is the ratio of SNP het to HOM ratio across all 3,000 samples, and you can see that our rates are essentially 0 compared to the existing call set. Go ahead.
A quick question on it. So, I was curious on the neural network architecture.
Sure.
This often requires a lot of hand tuning.
Yeah.
So I guess 2 parts. How much hand tuning of the architecture do you have to do? And was there any domain knowledge, for example, gene boundaries or anything else?
It's actually totally embarrassing. It works incredibly well for every type of image recognition model we have. So if you go to the deep learning code base, you can train it on ResNet 50, you can train it on ResNet 101, you can train it on Insertion V2, V3, Exception. The only one that I know that doesn't work super well is MobileNet. MobileNet, because it's designed to have less parameters, it doesn't seem to be able to perform as well as any of the others. And we've also applied things like the AutoML approaches that Google is working on, so we can tell the machinery to just figure out model architectures for us. And those work pretty well too. So it pretty much works. It's actually kind of embarrassing. We could have chosen any model. We weren't clever in choosing Inception. It was just available. We could have chosen ResNet. I could be telling you all about ResNet models. But literally, these results are from an Inception V3 model.
Well, I mean, if you actually use Go ontologies as an addition to that, you can possibly get the function.
We could, yeah. I mean, we have not done many experiments with how you might supplement the data itself. So one of the reasons we push code out into the world, this is all open source, is that we're kind of hopeful that people will do those experiments. There's lots of cool things that can be done. I see there's one question over there.
One sec.
So going 2 slides back, where you had that kind of from the mouse, I mean human
Oh, yeah.
mouse,
Where is that?
My question
Here
was,
you go.
was this application of the entire model to the mouse or just transfer learning from the other model?
No, it's literally like you train it on human and then just run that model on mouse. Yeah, just pure inference mode. So it knows nothing about mouse genomes, it doesn't know about the data, and the thing is the data is super different too, which I didn't like really highlight, but like it's trained in this case on 50x 2x148 high-seq 2500 data, and the mouse data is 27x 2x100 GA2 data. And like we don't attempt to account for any of that.
And all your work is in terms of casting the stuff into images and then working with
It is, but I'll talk more about that in a bit. So we're way beyond images at this point.
So you're not doing any transfer learning in any of your work, right?
This is sort of semi-transfer learning. This is like a little bit of fine-tuning. So originally we started from ImageNet. So we actually took the model that was pre-trained for dogs and cats, and then we demanded that it specialize to genomics. And then these results are starting from our general Illumina model, fine-tuned against these data types. Now we just go from scratch. But that's largely an engineering issue. It's kind of annoying to worry about restoring model variables all the time and things like that in TensorFlow. So we have so much data now that we just go from scratch. I think there's one more question.
Can you go back one slide?
Sure. This side?
One more.
This one.
What's the genome build?
Here? Oh, you mean here? Oh, I see what you're saying. You're saying what is this thing?
Right there, yeah.
Yeah, so this is boiling down to, the National Institute of Genomics basically maintains an official version of the human genome. And every few years, they like rev it. So there's a version of the human genome. So the current version is called B38. It's like roughly 38 editions in. Every few years, like they basically fund people to go out and say like, fix this area on chromosome 6 that we can't measure super well yet. And so people go and get grants and like go and study that area until they know exactly the base pair resolution of that area. And then they tell the Genome Institute, this is what the sequence looks like there.
For one specific person.
It turns out that a human genome reference is like a weird pool of about 10 people. So there's a tube, I think, somewhere. There's like the original material, which is based on 10 people's genomes.
So this is always sequencing the same 10 people.
Every time you're working on the human genome reference, like that's what you're talking about, is this weird like hybrid genome. So it's unclear, and actually some of the choices there have nasty implications analytically for, if you're studying Neanderthal or chimpanzee, it's not ideal to have a mosaic genome as the reference in humans, because it has different properties. But roughly, yes, there's a tube somewhere that's the official human genome reference. People study it extensively over time. And there's an entity that periodically like snapshots all those differences, incorporates them, and does a new release.
I see. And then all of these models, the purpose is to predict what, like, is to find the errors and to predict all of these.
Yeah, so it's really to find all the sites that are genuinely different between the reference genome.
I see. Given like a billion reads or.
Exactly.
Okay, I see.
Because this is what happens. Like if you go pay $1,000 to have your whole genome sequenced, like the instrument gives you a billion 100 base pair fragments, and then you're not really interested in that, right? Really want is like, hey, here are all the different, here's my genetic variants, and what do they mean? And so you have to sort of jump to that problem, and this is a technology to give you that answer. Good, so what else we got? So yeah, so we open sourced this thing. So we did a lot of work for many years, and we figured out, okay, we should probably open source it. So we pushed it out on December 4th, 2017. There's a plot of our number of stars on GitHub. So this is all the other major bioinformatics tool. I wouldn't be showing you this slide unless this was the big green vertical bar was us. So we were basically the #1 bioinformatics tool within a week on GitHub. Super exciting, lots of people talked about it. There's Eric Schmidt talking about it, and Sundar, and it's in the clinic. So one of the things that's interesting about this field is like, people are being treated because they have major medical issues that you can learn about in genomics, and like the tools that make errors today are in widespread medical use. So like if you do a better job, these things get incorporated into the clinic and start treating people immediately. So within 1 month, this thing was already live in a medical genetics clinic in Norway. So that's exciting. So people are really using this thing. And there are lots of people who are looking at it. So DNAnexus is a startup, it's a Google Ventures backed startup, but it's down over on El Camino in Los Altos. They basically provide a cloud-based platform for running genomic analysis. So they do all these analyses all the time to sort of establish how well how good tools are, which tools should they recommend. You can see kind of their assessment of the performance of the open source version of DeepVariant, very accurate. In this sample, very accurate. And then they did this sort of stress test as they introduced more and more errors. And what I love about the actual slide I meant to include is that this data set, well, anyway, I'll skip it. But there's a better version of this slide that talks about a more interesting data type. But unfortunately, it's not there. Okay, so that's like DeepVariant in a nutshell. Like, it's an interesting problem. I hope it makes sense why we work on it. Now I'll really explain how it works. So now is a good time to have questions about genomics before we start getting into the math. Go ahead.
So out of the errors that DeepVariant makes, what is it due to the sequencing errors, or is it like avoidable? And compared to GATK or SAM tools, what kind of errors does DeepVariant avoid?
So by far it avoids, yeah, I would say, the best answer to that will come in a second. I would say, like, for the errors, like, this section explains why we make fewer errors. But I think the takeaway really is a question of, like, how good could we possibly be? At some point, we'll be sequencing data limited. But I would guess that we can get 2 more 9s in accuracy. So we could be at less than 1, maybe 10 errors in the whole genome before we're really data limited. It's still analytics limited. Like, you could look at this and be like, that was crazy, why did it do that? Go ahead.
I just wonder if you're interested in applying the method to cancer mutation calling.
Yeah.
It's a slightly different problem. I would say it's different. The training set must be very different.
Yeah, so we're actively working on that, I would say. The problem is, how do you get training data? It turns out there's not that much good training data for cancer genomes. Good, I think
so correct me if I'm wrong, but basically you're asking your model to output 3 different decisions, right?
Yeah.
So you're looking at particular variant, like particular part of the genome.
Yep.
So if you make a more complex system where you want to look at different parts of the genome and ask your system, so what would be the combination of these variants? So you'd have to output, I don't know, hundreds of different variations. When do you think there's going to be a limit when the model is going to start making errors?
Well, I think, one of the things that we were nervous about a little bit after this work was like, how many categories can you learn effectively? So another problem we work on is like species classification, because it has a lot of species. So I can tell you do these sort of techniques to 13,000 categories pretty straightforwardly. It turns out actually that the implementation of the softmax, which is the thing that drives that, actually becomes a bottleneck at some point. And so it actually becomes an engineering problem. Like, how do you do a softmax over 1 billion categories? It's an interesting problem on the engineering side.
So question. Quick question. You probably already mentioned, how many genomes do you use to train your model?
Currently we train, I think we use 8 replicates of N12878 from Verily, plus a variety of genomes that we've downloaded or people have given us now. Probably, I bet you we're probably up to 20 to 25 genomes, each of which we downsample extensively. So we train on the full data set, the 0.9 downsample fraction, 0.8, 0.7s, all the way down to kind of 0.1s. But honestly, we could train on much more. I think currently, our training set size is roughly 100 million examples. But we're training limited at this time. So when we train DeepVariant, we train it on 25, usually V100s, already in parallel for 3 days, because we want to get to roughly 30 million steps at a batch size of 64. But if we are using TPUs, which is the Google kind of alternative to GPUs, we think we could probably train on 5 billion examples in a day.
Cool.
So that's coming, and that's quite exciting. We're pretty certain that'll give us another 9.
And for the metagenomics example that you were talking in the taxonomies in the species, how
Yes.
many do you use?
It's unclear. This is very much under development. So I would say that there's no tech. We mostly care about how quickly we can tweak that model. more than how much we can train it at real scale.
Thank you.
Maybe a naive question,
Sure.
Which is about the alignment.
Sure.
Is there a way of modeling a framework that one could imagine to build a problem that figures the alignment by itself?
Yes, there are lots of ways to potentially do this. I'm happy to talk after it. I'd love someone to work on that. I think it's possible. I think you can have a deep learning-based aligner that does potentially very interesting things.
Then the second question,
Yeah.
very quick, is would it be possible to create a fully convolutional model just to recreate from all these snippets that are aligned, a very clean signal on the other side, which would be like you input this image that you say you have encoded that you have like RGB, whatever, and then it creates on the other side of the model another RGB which does not have errors ever.
Yes, probably. You could use some kind of like auto encoder approach. Yeah, it would be very interesting. Error correction is a very interesting problem. In fact, that was the problem. I'll talk a little bit about that, but the example program, a library we released called Nucleus is structured for that problem exactly. So I'm not sure it was an image problem, but it was done read by read. But it's a very interesting problem as well.
I mean, it really does not matter how it's done in terms of implementation, but it's this kind of fully convolutional bottleneck.
Yeah, we should talk about this. It's very interesting.
Okay, thanks a lot.
Okay, so I think it's good. So why don't we talk about like how this actually works and like why it actually works. So this is actually what the data looks like. Now I've been showing images the whole time, but this is actually what sequencing data looks like. So this is the actual input for the program. These are each read. It's encoded in this crazy way. So those are the bases here. You can at least see the kind of the bases. This weird looking string there is an encoding of a probability of like that base being wrong. And there's tons of metadata. It tells you like this read is on chromosome 20. It's 10 million bases over plus 4,154. Like that's the actual data that the sequencer spits out. You've got a record. You've got a billion of these things. So you got to align it. These are already aligned. And really the challenge is go find all the real differences. You need to do this at like roughly 1 in a million error rate. And this is 5 data points of the 64 reads that span just one base in the genome. So this is really like big data problem. And it's pretty grim. So this is really hard. Like nobody can possibly spot the SNP here. It's impossible. Somewhere in those reads, in those bases, is something that's really different. Once you visualize it, it's like vastly easier to see what we're talking about. So it turns out that this is a deletion, and this is actually an insertion. So these are like insertion markers. So what's happened here is this base has been deleted, and that in another chromosome, they've gotten 2 additional bases at this exact same site. That's how you read this. So you see half the reads have these 2s, saying that things are added, and one thing was deleted, actually. So zoomed in that you can see it. And there's a nearby genetic variant here in the G. So it's highlighting, but I should probably explain what this is. Each one of these bars is a read from the sequencer. That thing on the top is sort of a histogram of the differences from the reference. So you can see in the point where it's G, it sort of flares out because it's colored differently because it sees so many Gs. Each one of these reads has lots of errors. This is the errors I'm talking about. Like this thing is, I don't know what this is. There's some weird looking C here. There are a whole bunch of these weird artifacts here. What is this? These A's aren't there, it's not easy to figure out what's really going on. And so one thing that you would do is say, okay, well, like, how do I think about this problem? Like, what would I look for? There's all these hallmarks of like being real and not being real in this data. One very clean hallmark is that the G is in phase with the deletion. Because the read reads across one chromosome. So if they're really genetic variants, that this deletion is real and the G is real, they should either be there both or not. And you can see that behavior. See, everywhere that's 2s don't have Gs. And everywhere that has the deletion has the G. Sort of a hallmark of not being in error versus this is like, well, I don't even know what's going on there. So it has all these sort of properties that make it say, hey, that's why it's weird, that's why it's not weird, that's why it's real. So how would you solve this problem? So the normal way to solve such problems is to start in the simplest possible way, which is just count. And counting works pretty well. And programs exist that count. So there's a program VARScan. This is still widely used in the cancer space to just count up differences. And you can build a threshold. So the example is you say, hey, if there's more than 5 differences, it's real. They're all consistent. It's totally reasonable. And this works really well and has like roughly a 95, maybe 90 F1 score, if you go this way. So it's not bad, but it's not even close to like 6 nines that you might want. So what would you do if you want to be a little more sophisticated? So the next sort of upgrade you can do in this is to move to a Bayesian framework. So what you're doing here is now you're saying, oh, I have some Bayesian model. What's the probability of being a variant given my data? You invert it, and you compute all these things. I won't walk through this, but you basically ultimately end up with like a likelihood ratio test for like 2 different hypotheses. And that gives you a statistical framework where you can think about things like significance testing. So it's like, oh, if I'm more confident than 1 in a million, I'll believe it's real. And you can incorporate things like priors. It's super attractive. And the challenge, though, really in all of these models is this nightmarish problem of what's the joint probability function. So almost always you kick the can down where you say, oh, I assume all my observations are independent. I'll multiply all the probabilities through and I'm done. So this is your standard kind of Bayesian approach when you're starting from multiple observations and you don't know how they correlate with each other. So you just assume independence and go. So like naive Bayes, the classifier, is basically an encoding of this. So what is really going on? Like what is the counting and Bayesian solutions really see? At this G site, you can think about it as like you're looking at one side of the genome, independent of everything else, and you're just counting. There's sort of basic frequentist or Bayesian statistical integration. So you can really think that I don't even get to see anything to look on the left. All I can do is a big pile of these things over here. I say they're G's, and there's some that are not G's. And I can compute from that, like a count, I can compute their probability estimates. Like that's fundamentally what is happening in these models. These work okay. I mean, they're, counting is good, Bayesian stuff is better. The more sophisticated thing you can do is basically go to what's called a Markov model, which is an attempt to allow you to see when you're looking at the G information nearby. Because you want to take this context into account. The thing I talked about with the twos versus the deletion, like that's obviously material information. Like completely ignoring it's a bad idea. So what you do is you build a hidden Markov model that sees over the whole read and sees a reference. And it can compute all sorts of things. And I won't bother reading this, but this is how the GFK works. At the end of the time in the GFK, you basically got to this paired HMM model, and this is what it sees. So you say, I get to see each read. So it's better than a big pile of random bases in isolation. So you see all these reads together. Each read you get to see as context, but you don't get to see them together. So you get like horizontal context, but not vertical context, right? Like that's what you're giving up. An HMM per read sees only the read. It doesn't see the other reads. It's unclear how would you incorporate that data, right? So you ultimately end up horizontally, you see horizontally but not vertically in this image. The thing about, and the reason deep variant works is simply just that convolutions are the generalization of that approach vertically and horizontally. So you have multiple convolutions, in particular in stacks. So convolutions feed into other convolutions, expanding their visibility as the stack of convolutions go up. Like in an Insertion architecture, it's super deep. It's like 11 layers of these things, each of which is like 3 units. It's like very deep. So what's happening is you have these like 3 by 3 comms. So this is seeing 3 bases by 3 bases, 5 by 5. You see that it starts to see the context. And the more they have context and the higher these things are, the more you can kind of incorporate all the information. So that's all that's really happening. Deep variance is just in a series of these straightforward steps from counting. You count, and you want better statistics, then you want to see horizontally, and then you want to see vertically, and the only way to see vertically is really probably to go to a convolutional approach. And then the thing that's nice about the machine learning is that it figures out how to put all the weights in here, because that's the thing we don't really understand. We would be very happy to write better than naive Bayes algorithms if we just knew what those terms were. But it's very hard. So at the end of the day, what's interesting, deep variant really has one parameter, and honestly, we're seriously considering deleting it. So there's one parameter. What is the probability that that the difference is real? And the reason that works is because you can compute things like this. This is the probability of being real as estimated by the program, which is basically the statistical model's probability or confidence, versus the empirical error rate. Because you bin things up, you say, okay, everything you said that we were this confident in, like you have the right error rate. And this is in this weird Fred quality scale, so you can think about this as, this is an error of 1 in 10 to the minus 5 rate. So you take everything that you said you were that confident in, and you just compute its error. And what you get is in like the naive algorithms that use like Bayesian integration but do naive Bayes style independence, they're super overconfident because they can't account for the joint probabilities. But deep variant learns those joint probabilities, and so it gives you a good curve, a good fit on this perfect calibration, and then you slice it wherever you want to be confident. If you want to be super, super confident, only take that stuff. If you want to go into the bottom of the barrel, you can come down here. And it turns out that actually now it's just, there's no threshold that really moves anything anymore. It's so good at classification that like almost nothing is ambiguous. It's either right or wrong. And then it makes terrible errors that it's super confident in. And those are things that we train more to get rid of.
Why is this continuity in the same spot?
Where, oh, here? This is a hard-coded this is a parameter in the GTK where it switches to a different version of the binomial calculation.
You got it in the same spot.
Where?
You vary it.
Here? This?
Yeah.
Here. Oh, this is just this is running out of data. There's almost no data in these bins. You know, this is calculated over something like 100 billion data points. But even there, the problem is that, you know, this error rate in this bin is implied, you know, 10 to the minus 6. So you have to have a lot of data to collect the proper statistics on that rate, and so you kind of turn over. We had more sophisticated versions of this calculation that have a Bayesian model behind it to try to incorporate the uncertainty, but that was too much. So, and this is true for all sorts of different classes of things. You know, this is why it's working. And you can see the alternatives are kind of behaved in a non-standard way, with weird discontinuities in them, with weird shapes, etc. Okay. So I think what time is it now?
1 hour. 1 hour and 3 minutes.
Okay. I think I will finish with this slide then. So one thing I just want to talk through is like, why do we like this approach? Like, what are things that people can take away? So it's sort of weird to talk about images. Like, we're taking genomics data and we're turning it into images. But I just want people to understand that like that's not such a big deal and it's not so weird. So this is the original deep variant V1 encoding. So these things were really RGB images. And the actual encoding is the red channel tells you which base you are. The green is encoding that probability estimate. The blue is the strand, and the alpha is this weird, like, does it match the genome or not? And it gives rise to images that look like this. You know, green, the bandit shows you what bases are there. These like highlighted images is because you don't match the reference, and so your alpha channel is different. And so you can look at this as a human being and be like, okay, I can kind of see structure, I can kind of see the variant is in the middle there, this kind of glowing band. And it worked okay, and then you just fed this at the image interpretation system. In large part, this is because this was pre-TensorFlow world. So we were trapped in disbelief, and disbelief basically worked in RGB space with completely unmodifiable version of Insertion v2. So you have to shoehorn your data in. But if you think about it, it's not that weird. So you have an image, and this thing that I was showing you earlier is just this is a nicer representation of the data, but you have a width by height PNG image. This is really just a width by height by 3-channel tensor, where the individual values are allowed to scale between 0 and 255. So you know, it's just a matrix of data. So once you realize that, there's no fundamental reason that you can't think about it as a width by height by n-channel data tensor. I mean, you're really steps away from it. You go from 3-channel image to an n-channel data tensor, and the you know, if we had started calling it a genomics data tensor, people would be like, wow, those guys are super clever. But we called it an image, and they were like, genomics data, why would you put it in an image? That's crazy. They're really the same thing. And in fact, the latest versions of deep variant actually are now 9 channels, I believe. So this is the 6-channel version. The open source version is using this representation. So instead of shoehorning things into this image, we now are directly encoding individual channels with specific representation. So the read base is now in channel 1, the quality is in the 2nd channel, the accuracy of the mapping on the genome is another. You can see which direction the read came from, whether or not it supports the variant, and here's that like very clear, like, does it support the reference genome or not channel? And so now in the open source version of the variant, we're just like adding channels. You can add any information you want. You know, when you think about adding interesting channels, like are you in a gene or not? It's pretty trivial. Just put it on the back. let deep variant project the convolutions through it, and you're done. So we're quite excited for the kind of things people do. Like, we do a lot of this kind of representational exploration in Google. You know, should we one hot the read basis is an active question. You know, how big should the image be? It's not obvious. Like, it should be this big. It's 221 by 100 bases. Maybe it should be 215 by 80 bases. Deep. Oh, maybe it'd be better. But these are all becoming kind of empirical accuracy questions. And it turns out that moving from the PNG representation to this more separable data representation is a huge performance gain, because the model sort of gets cleaner data. So the version of deep variant that won the Precision FDA has an F1 score of 99.3. But the open source version, the very first release, was at 99.84 already. The current version that is releasing is like probably another 9 or 2. And that's, you know, it's useful to say, like, open source release was December 4th of 2017. We're 3 editions in, and we have like another 9 or 2, simply by training up more, playing with the data representations. It's just like fundamentally impossible if you're like hand tuning all these parameters. So we collect data of different types, we're doing badly, incorporate it, and it just gets better and better and better. Good, so I think I'll stop there in the interest of time. These slides, I believe, are available. Certainly this section is available in the TensorFlow Dev Summit YouTube videos if you want to see. But anyway, there's a library to sort of make it easier to do this kind of work, in case anyone is interested. If you want to see this part of the talk, there's one from my colleague Corey last Friday, sitting on YouTube right now. So thank you very much for the invitation. Sorry I didn't get through all the slides. I warned everyone up front. You might not get there. Thank you.
Maybe we have time for a few questions.
Oh yeah, I'm good. I just don't want everyone to feel trapped.
I think
I will not be offended if people need to get up and go.
If that was a shoehorning, that was a very elegant shoehorning. Okay. What if I just stand here and then you guys can maybe line up if you've got questions?
Hi, Mark. Would you mind mentioning some of the other projects that you're working on?
Sure. So we're very interested in this nucleus project.
Okay.
So this is our mechanism for getting other people empowered to do genomics work in TensorFlow. So this is materially improving. And this actual library is like what drives deep variant now. So we've ripped deep variant's core engineering infrastructure out of the code base, put it in a standalone library so people can do it. So this is a pretty active effort. We have a lot of open research problems, one of which is in the metagenomics space. Okay. So we're quite interested in how to do that. In general, we're very happily working on lots and lots of problems, not all of which I can really discuss. But as things come out, I would say like there will be many cool announcements over the next 6 months as things progress, because we have projects that are maturing. And then we'll be out giving talks about them and disclosing how they all work in intimate detail.
What is your inference on what hardware running on?
This is a great question. So this actually has systematically been a problem. I like it. Not everyone on my team likes the conflict. So there are 2 basic criticisms of deep variant that are running around. One is that people don't think it's a big deal to do this because they're like, oh, you can call variants GDK, and you're like, wow, this is a big pain. So we fight over whether the additional accuracy is important. And then we fight over performance. Right now, deep variant is pretty slow, which I like. Because the argument that it needs to be fast is sort of on the wrong side of the technical trajectory. So when we report, we tend to run deep variant in the maximally accessible configuration, which is like AVX only built instructions for like no higher than a Broadwell, assuming no lower than a Broadwell class Intel processor. And when you run on 64 cores, the inference step of deep variant alone takes like 11 hours on 64 cores, which is pretty slow. I mean, it's not so much slower than any other program out there. I mean, the other programs are all super fast, but this is not true.
And then 11 hours doing the prediction or the training? The prediction only?
Just the prediction for one
Got
genome.
it.
But that's on a CPU-only configuration that's suboptimal. So if you want to move to like a K80 GPU for inference, this is moving to 8 hours. If you're going to use a P100 class GPU, you're falling to 2 hours for that operation. And every time you do that, hold on, let me just. Those are the actual numbers. So the actual runtime on a K80 with 8 CPUs is 7.5 hours at $6 for the inference step. And then if you want to run a P100, it's 2 hours and $4. And the TPU numbers, I would say, are extremely exciting, and we look forward to announcing them. But yes, this is, and it's just used to say, like, where a lot of the action going on in silicon, it's in these GPUs and TPUs. So like, I don't want to be, we make the CPU numbers accessible because like lots of people have on-premise compute and like, you want to run on a CPU. But if you're trying to get high performance and low cost, you're going to a very sophisticated model that sort of almost demands cloud-like instances. Like you don't want to invest heavily in a P100 fleet because there's already probably a P120 that's coming out in some time, right? So for us, like the biggest attraction is that like, we can put out numbers that are like $50 a sample in a trivial configuration, but we offer a version on cloud that's just running in Dockerized containers in something that looks a little bit like Kubernetes, so it can be robust to preemption, and it uses GPU, and that thing costs roughly $5 instead of $50. And we can just hang out and wait, and our stuff gets faster and faster. And since it's embedded in TensorFlow, TensorFlow is obsessed with speed. There's not that many people who are like lovingly optimizing their genomics pipeline, but our by far heaviest lift is in TensorFlow. And there are a lot of people who care a lot about TensorFlow inference performance. So like we literally can just wait and we will be the fastest, cheapest thing. Because our competitors in many ways are sitting at like $2 to $5 a run, and we're close. And every 6 months, look at this jump from a K80 to a P100. It's huge. The next round of these things is just cheaper and faster because ultimately, like, we're kind of on the technical trajectory of the pixel phone development in a weird way. But ultimately, that's what matters for cost. Thanks.
More questions back here?
Perfect.
Okay, so just for a minute about this one, we should add the row about B100. We should double your performance, but of course I cannot guarantee that.
Yeah.
You recognize technology.
It's great. Yeah, I mean, it's great. Go hang out. Things get faster and faster.
I have two questions.
Sure.
Maybe a little bit more about implementation.
Sure.
Have you ever tried with a true convolution, which will spare you this kind of ill-posed pooling step that you must be doing right now?
I would say yes and no. So we've done like neural architecture search in an attempt to solve the problem. The deep variant data is like a stock problem for the AutoML efforts at Google. They pass through like kind of pure convolutional problems. I have to say though, we've never trained like a very clean, simple model architecture. It'd be super interesting for someone to do this. It's possible that it would do well. Like one of the things that we would like to reach is a state where we think we are parameter limited. So we don't think we're not fully utilizing all the parameter space right now of even Inception v3. We know that if we keep letting deep variant train, it keeps getting better. So it would be nice to have it actually flatline so that we can tell the difference between a model with more parameters. So that's one reason that we're excited to get done to TPU-based training of 5 billion images, not 100 million, because we want to see what the actual architectural limit is. I don't think we're there.
And then there is another thing that I didn't really get. So when you do one prediction, you are putting in one image, or you have to go through multiple images?
In the normal case, it's one.
Okay.
So you have a candidate variant, you get a data tensor, and you run an inference. It's slightly more complicated in the scenario where you have multiple possible candidates at the exact same site. We actually split those all into multiple images. So we have one for allele A, one for allele B, and one for this weird hybrid AB allele. And then we merge those all back in. It's not clear that that's a very good solution.
So that's why you need all these hours to run it. So that's the only reason. Because one pass through VGG or whatever ResNet should be milliseconds, right?
Oh yeah, exactly. For us, one run of this thing is in an Inception v3 evaluation at every candidate variant in the whole genome. So we're running typically between 7 and 10 million inference steps for and an N of 1. So people like this because this is a weird configuration problem for deep learning. You're like, so how big is your inference problem? And you're like, well, it's pretty big. You know, I gotta get through 10 million images as fast as I possibly can. So it really stresses out the I/O pipelines and things, actually. Because we don't care about latency, we care about throughput. And we care about cost efficiency, which is, again, a little bit weird in the deep learning space, which is one reason that we like being at Google, because we're kind of an interesting, weird object over there that's stressing the pipelines in strange ways. So.
Many thanks.
Okay.
Hello, thank you for the amazing talk. I think you touched on this a little bit. So, can you elaborate more? How do we use deep learning to classify disease or cancers, and how do we solve the finding the training data problem, and how do we increase number of N?
I would say it's unfair. It's really unfair how to, you know, if you're asking like how do we use deep learning to say model disease variants, like I've yet to see any work in this space that I think is really compelling. You know, the problem is right now the largest kind of cohort level data sets that you could possibly get your hands on are for things like height, and you might be able to get like 1 million samples where you know their actual heights. 1 million samples is not a lot, because you know we know even with basic statistical techniques that like we're not even close to the number of, we haven't found all the height variants, let alone be able to model your height accurately from your genome. So I think this is many, many years away, unless you can figure out some like super clever way of boosting your data or reducing the scale of the problem. But as defined by like genome-wide association studies, like this is super challenging. Yeah, so I think there's that. I guess, you know, on a positive note, I would say that I am optimistic about people's ability to use deep learning models to help curate the literature in clinical genetics. Because, so there's one thing for like height, this is a very complicated trait involving like 10,000 parts of your genome. It's another thing entirely to have like rare genetic diseases. These are often well characterized, some single gene. The problem is the characterization of that is like in a whole bunch of databases that are kind of like free text. And that's something that deep learning is pretty good at now. So I do actually think that will help a lot, this automation. And if people want to get involved in that, there are multiple Kaggle competitions with exactly that problem. Like the variant interpretation from literature is well understood as a proximal deep learning problem in medical genetics.
So, have you found any specific cases where deep variant doesn't work compared to GATK or other methods?
There have been a couple of examples. So one scenario, and we'll have a blog post about this, so in the next couple of weeks, we're going to do another launch of deep variant, and we're going to talk about some of these cases exactly. So if you want to hear more about the details, you should keep an eye on the blog. But at the end of the day, there are two cases that I can definitely think of. One is for exomes. So we train deep variant only on whole genome data. But a lot of people have exome data, so this is data that's captured just on 1% of the genome. It looks pretty different from a normal genome. The 0.5 release of deep variant was largely targeted at improving the accuracy of deep variant on exomes. And we went from like 0.93 F1 to like 0.97 F1 simply by just training it on some exome data. So that was pretty attractive. That's now the best exome caller that we can find. We outperform, I think, everyone. The other area where we've done notably badly is PCR positive data. So we trained only on PCR free data because our view is that like that was the future of genome sequencing. It turns out for multiple reasons. One, historical data sets exist that are PCR positive, and people are starting to do more PCR positive data again. So we grabbed a whole bunch of PCR positive data and are now radically better on that. So if you want to see the example of that, like there's a nice post by DNAnexus evaluating deep variant, and where the only case where we don't really do super well is on this PCR positive data set, where we're a little bit better than everyone else, but not by a lot. And I think that'll change soon. It's nice, we can just say, okay, well, what kind of data is weird? Okay, well, let's just download that data and retrain it. And it gets better in every scenario. It gets better at the data we've been evaluating, and it gets better on this new data.
I did my PhD in biochemistry, so I know a lot about things that you already told. And now my business is in doing hackathons on different things. So my question is about genetic data. So how much data do you need to analyze data of cancer patients to then use it to correct mutations?
To correct or
yes, first to label data, then to give it to companies, for example, or whatever. So this process. So how much data you need of these clients, of these patients, because you told that it's not enough now.
Yeah, so if you're asking like, could, you know, what kind, there's some, you know, classic formulation of this problem of like predicting, you know, what type of drug you should give someone for cancer, given that they have a cancer. Is that the problem you're envisioning?
No, you're working the data of patients, yes?
Okay.
And you are searching for mutations.
That are related to their cancer.
Yeah. Because one of the guys asked you a question,
yeah.
And you told that it's not enough data now for you to analyze them. So how much data you need?
It's unclear. And basically, it boils down to the problem. Cancer is different from germline. I should just be clear. Like germline is very hard compared to cancer because the vast majority of the traits in your germline are not like determined by one or two things. Like it's determined by thousands of variants, and the total amount of risk that they explain is not enormous. Cancer has much bigger effect sizes. Like you have a causal mutation, right? In like, or really an accelerator mutation, like HER2-positive breast cancer has a very particular type of amplification. Knowing that amplification tells you a lot about the treatment course because it's a huge effect mutation. That said, most cancers aren't driven by a small number of things. So if you're asking questions like, how much data do I need to identify like key driver mutations? Probably
or some new mutations, for example.
Well, calling new mutations is relatively straightforward. Interpreting their clinical utility is a whole other beast. So I would say I'd like to see millions of patients. I would start to feel good about millions of data points. If we were going to make serious decisions about people's cancer treatment based on pure analytics alone, you would really want to feel super good about this problem. Otherwise, you know, the value, and I think a lot of companies are making a lot of progress already in this space, but they do that by leveraging enormous priors, right? You can think about like the literature, the drug treatments that are recommended. These are giant priors. Like, to rediscover what you should do just from the data alone requires a lot of data. Like if you wanted to classify the 3 types of breast cancer, like HER2-positive, EGFR, and FGFR, I think is the other. Like if you wanted to even figure that out without seeing anything but people's cancer genomes, this is pretty challenging. You've got a lot of potential mutations to look at. And you need to kind of average away the noise in some way.
Thank you.
Sure.
Earlier on in the presentation, you said that a deep learning-based aligner would be really interesting. Could you explain what you meant by that and why?
I think it's super interesting, one, because database lookup methods are a pretty broad class of things that we do in bioinformatics. So like BWA and BLAST, and like all the things that align like protein sequences to databases, these are all essentially the same thing, right? I have a giant database of sequences. I wish to jump into the database and find things that are similar, right? Like that's fundamentally what you're doing. And the similarity algorithm is now O of n squared, Smith-Waterman-like algorithm that computes kind of pairwise distances. If you need to be faster than that, you like hash in and then do it. Like that's fundamentally how all these programs work. You like hash into the database based on k-merge or something, and then do a more expensive algorithm to compute a score. The nice thing is, the reason I think that's exciting is one, those hashing algorithms are ad hoc, and we know that in general computer science data structures, deep learning is starting to replace them. There's a beautiful paper by Jeff Dean on this called Learned Index Data Structures, if you want to see this. It's about like, can you build a B tree where the key is a deep learning function? Can you build a hash function that uses deep learning to hash? Because it's very clear, and they show that it's possible and that it has real world advantages. So one, you might be able to learn how to index genomic data structures better, which would be super interesting. Two, you might be able to, so there's all sorts of optimizations you can imagine doing. The other thing that's kind of cool about this is that at the end of the day, you're generally not getting perfect data, right? Like, it's one thing, you know, like string matching is a pretty well understood problem. So doing like Burroughs-Wheeler transform on your genome and then indexing into that data structure is really, really effective if there are no errors. If there are errors, now you're in the ad hoc algorithm world where you're like, oh, I'll do some weird tree search algorithm that will allow 2 mismatches and this type of deletion. And like, so you have these giant parameters that get all tuned up to sort of figure out how to be robust to error. The deep learning stuff kind of doesn't care about that. So my favorite formulation of this problem, which I would love somebody to do, is I would love to try to do a prediction problem, take the human genome, take reads, and try to predict which bucket it goes in the genome at different resolutions of the size of the buckets. So do 100 base pair intervals, do 1,000, do 10,000. Can you learn how to jump into the genome? It's like a hashing problem. I bet you could do that. I bet you could be super robust to errors.
That is the metagenomics problem that you were talking about earlier. That's what we do.
You're doing this jumping into the genome?
In the metagenomics, that's what we do actually.
With the deep learning approach?
No, not with deep learning, but the traditional methods.
Yes, so the K-mer, like if you're going to use K-mer methods, these again, you have to do some kind of search for the error. I think you can learn how to do this problem extremely well. I would love to see kind of a deep learning-based replacement for K-mers. Super interesting. Sure.
Okay, so as I understand it, one of the goals of your group is to find applications of machine learning in for genomics.
Yeah.
Solve problems in genomics using machine learning techniques.
Yeah.
And you've done that with these techniques from deep learning.
Yeah.
What about techniques from reinforcement learning? Have you seen any good applications for those? And do you have any in the pipeline?
I say yes, and I can't discuss that. That's how I would say that. So I think it's super interesting. Reinforcement learning is wildly interesting. There are lots of things that would be interesting to try to do with reinforcement learning and bio.
Anything actionable?
I say we have lots of ideas. We're trying to figure out what to do. But I do think this is very interesting. At the end of the day, there's lots of cool problems that could be reimagined as reinforcement learning problems. Tractably, yes, I think. I don't know. We're still thinking. But I really can't discuss too much about those. Those are very early stage ideas.
Okay.
Sure.
Let's take one more question and then we'll thank our speaker again.
I don't know if we can talk about this, but is Google working on using deep learning to applying it to gene expression data, like RNA sequencing and, you know?
This is interesting. So I would say it's been one of the things that's been on our mind, like, and we're not working on it actively. So it'd be very interesting if somebody wanted to work on it. I mean, you could look at it this as 2 kind of formulations. You could look at like trying to, you know, if you take a program like Callisto, for instance, which I think is a pretty sophisticated RNA quant program, one, there's like pseudo-alignment, which might be solvable through deep learning approach. Could be interesting. 2, it's given that you have aligned reads and all their ambiguous placements, it's not obvious how to compute the actual expression from that data, right? So right now it sort of counts, and there's some like likelihood function, a bunch of people, some of whom are at Google and Brain now, like wrote out, right? So if you look at like the likelihood function of Callisto, like there's terms for amplification, poly-A, decay, and all these things that are in there. It's probably not as well as you could do if you had a deep learning model there. So the question there would be kind of, how do you get the truth data? But I bet you could build a deep learning model that was way better at taking read counts, or the actual reads from RNA-seq aligned to a gene and estimating its true expression. So I think that's a super interesting problem.
And pertaining to that question, so like the gene expression, it has the same problem as like there are not enough samples with, you know, with gene expression. Say there are cancer patients, there are maybe 100 samples. Do you know of any way to, let's say, augment the data, or you know, applying some generative model with deep learning, that sort of thing, to gene expression data, or any data with limited sample genomic data?
Well, one of the things that's nice is the reason the cancer problem or the germline like, disease problem is hard is not because there's not a lot of genomics data. It's the labels that are hard to get. People generally don't want to share medical records. Like this is the most protected kind of data we have. So it's really the N there is observations of people with particular height or particular disease, because your N is like of the disease states. And so it's very hard, like data augmentation, like what does that look like in that field? But at the technical kind of problems like deep variant or RNA-seq, like the original version of deep variant, the one that won the precision FDA, was trained on one sample. So the N of 1 is N of 1. It's just that we recast it so that we got 5 million training points in that genome by taking every candidate and every candidate variant and treating as independent. So similar tricks can be played in RNA-seq. Like you could quantify each gene separately. You can downsample the gene. Like it has the same true expression level, even if you had half as many reads. So you have a lot of those tricks. I think you can get a long way with those tricks because the bar is pretty low. I mean, you know, the likelihood functions people are writing are not that sophisticated. So, I mean, so if you're asking like, can I do better than the existing tools? Probably quickly, and I bet you could do that with a small N. And RNA-seq in particular has very large data sets that you can use. You know, the Geo database has like more than 1 million chip results in it. So there's a lot of data. It's just that you're not going to get labels of phenotypes off them, but you've got a lot of genes, you've got a lot of transcripts. And you can downsample and all sorts of things for the actual data. So I think it's totally tractable.
Let's go ahead and thank our speaker, but maybe that those of us who have questions can stick around and